Receptors of the retina separately. They are schematically drawn. So draw. They are located on pigmented epithelium cell. Pigmented epithelium. Epithelium. Rods and cones, they are receptor cells, yes? They are ends like rods and cones. Second layer cells, bipolar cells. We will draw several bipolar cells, neurons. They have two poles, this and this. This. Ой. This is first row of cells, second row of cells, and the third is the chain of neurons of retina. There are 10 layers, but the three main link of neurons important. And here we have the multipolar cells. Schematically we'll draw the big cells, multipolar cells, with their process. They are axons. Dendrite end form bipolar cells. But they are all axons goes through the retina and through the excavator pass. You must remember that if I will draw это cornea and fibrous coat. Inside we have what coat? Vascular iris. Here we have thickened part, ciliary body, and then choroidea. Three parts of the vascular. And down. Inner layer is retina, pars ceca and pars optica. No, ophthalmic artery come here, vascular, and optic nerve. And here lies behind that. Here lies lens, and opening is pupil behind. Pupil lens lies connected with ciliary body. Here anterior chamber, posterior chamber. Here fluid, anterior chamber, and here posterior, small. Lens lies. Thus in light rays pass through light refracting systems. What? Cornea, camera anterior, camera posterior, lens, vitreous body which fills here. What body? Vitreous body, lamina vitrea. Не lamina, corpus vitreum. Corpus vitreum. This. And pass onto the optic part of the retina, yes? And here we have external, from here, external, face to the choroid, pigmented cells, on which more inner lie these cells, then bipolar, then multipolar. And axons of these multipolar cells form this optic nerve. Optic nerve starts from these cells of the retina. Light rays, how pass to the retina? Pass so, in this direction, the light rays, and reach through all layers of the retina, through all layers come, reach the until pigmented, deep layer. Here it is more external, face to the choroidea. Rods and cones. Here, after simulation under light rays, iodopsin, rhodopsin, they discharge into the more small parts, and nervous impulse. After simulation, the nervous impulse appears. How light rays reach the rods and cones, and from here nervous impulse flows Light rays, so good. And nervous impulse appears. In what direction passes nervous impulse? In this direction. In opposite direction. It goes nervous impulse from rods cone, then bipolar, then multipolar, and the axons once more through the all layers, through the retina, through the vascular cord, through fibrous cord, optic nerve arise, the second pair, which through canalis opticus passes inside the skull. What part of optic nerve do you know? Infraorbital, infraophthalmic. Orbital, in orbital cavity, then in canal, optic canal passes. Here passes optic canal. And then in skull cavity we have crossed. Now the visual analyzer. You draw from all left half of the retina, you draw retina, yes? And right half of the retina with the same color, red, right and left half. To left, blue color, to right, right color. From this right half of the retina, fibers which arise. From medial half of this and medial of this. Inside the skull cavity, in sulcus chiasmatis, they are crossed and form chiasma opticum. Chiasma opticum.
Which sulcus?
Here we have sulcus chiasmatis, and sulcus prehae. In the groove, they cross in the skull. Chiasma opticum, only from medial side. From lateral side, they are not crossed and go without crossing. And what continuation it has? Tractus opticus, because it has another content fiber. Contains different fibers. What is this? This is optic nerve. Optic nerve. Word pair? Second pair. What parts it has? Intraophthalmic, orbital, canalicular and cranial portion. About 5 centimeters nerve. Until what part nerve is ended? In skull cavity, in chiasma opticum, nerve is ended. When the nerve is cut, atrophy is developed. What's happened? Nerve is congenital acquired atrophy may be developed. This blindness, of this on one eye. Impulses could not. It is like electric cable for impulses. It is not innervated eyeball. For innervation of during exam, when we say, please explain innervation of eyeball. What pairs of the innervation? It means sensory and motor. What nerve innervate? Motor. Motor. Optic is wrong. Very bad wrong. Mistake. Oculomotor, trochlear abducens, innervated muscles. Sensory innervation? Nasociliaris, lacrimal and frontal from ophthalmic of trigeminal. Sensory nerve of facial region is trigeminal. Trigeminal, the fifth, third, fourth, fifth, sixth. Sensory and motor innervation, and vegetative innervation of smooth muscles of the eye. But optic nerve, it is part of optic analyzer. Analyzer usually, all analyzers, what scheme consists of what part? The first, receptor, they receive impulse, produce nervous impulse. Then pass, conducting apparatus. Then subcortical center. And then cortical, where the analysis cortical center. Receptor conducting, where are the receptors here? Where are receptors? Rods and cones. Conducting apparatus starts from axons of the multipolar cells. Ganglionic cells, multipolar cells, ganglionic cells of the retina forms optic nerve. It is conducting apparatus. Only conducts nervous impulse, but not innervate eyeball. It is for conduction of nervous impulse which appear here. To conducting apparatus also. What part? Optic nerve, optic chiasma, optic tract. They belong to conducting apparatus. Where the optic tract is ended, what is geniculate bodies. What corpus geniculatum laterale. And by means brachium they are connected with colliculus superioris, yes? Superior collicle. Colliculus superioris, corpus geniculatum lateralis. Subcortical visual center. Do you remember?
Yes.
Corpus, name. Geniculatum laterale and colliculus superioris. This belongs to diencephalon, this mesencephalon. After crossing fibers, what fibers cross? Only from medial halves of the retina. But you must remember that our so-called visual field, from this part, light rays pass here, from this part, it is temple part, nose part, pass here. Temple, nose. And the same here, nose and temple. This and this. So fast. That is why when optic nerve destroyed, impulses could not pass and blindness developed. Chiasma opticum, for example, tumor in hypothalamic region develop. First feature that patients say that change their and some symptoms of destroy their visual function. What function is destroyed? Only in tumor of the hypothalamic region, hypophyseal region, which compressed only chiasma. This and this half don't pass impulse later. After optic nerve is formed optic tract. Why it is called not the same nerve tract? Because it has different fibers, here red and blue, here only blue, from lateral side of the retina of the same eye and medial side of the opposite eye. It is left, it is right. Tract. They have different contents of fiber. Tumor in this region, what parts of the field? How these fields check ophthalmologists? So they take a pen and say, can you see, yeah, or not, cannot. Then if this part is compressed, impulse, what part destroyed? Here don't see, temple, see, see, not see. These two. It is called hemilateral, bilateral hemianapsia. Hemianapsia. The two lateral sides of the visual fields could not see, only medial sides. Sorry. Bilateral hemianapsia on tumor of the region of chiasma opticus, hypothalamic region tumor, for example. Later on ophthalmology, but I give example that you must understand that different fibers cross here, medial fibers from medial part of the retina. After we have left tract, for example, destroyed the left tract. Here maybe, and here. Left tract, optic tract. Impulses from here come to the corpus geniculatum lateralis cells, yes? What's happened when it is destroyed? From what half of the retina destroyed? From medial nasal half of this eye, and temple had two right halves of the visual fields. This half and this half. It is called homolateral hemianapsia. Bilateral, the same lateral, homolateral hemianapsia. Gomolaterальная, homolateral hemianapsia. It is bilateral, it is homo, the same lateral. Side lateral. No, later in clinic you will study if you will be ophthalmologist. Check. And on retina we have turn projection of the fibers, head down projection. After this optic tract, it is ended on corpus geniculatum laterale. What is this? It is subcortical center. Receptor, roots, cones, and also here participate bipolar, which send from roots and cones to the multipolar cells and multipolar. They together form receptor apparatus until optic nerve, optic chiasma, optic tract, corpus geniculatum, subcortical center, corpus geniculatum laterale, and colliculi superiore, and cortical center. From here, after this shape of fibers, they not directly go to the cortical center, lies in occipital lobe. The occipital lobe of the brain. Here we have what groove? So on medial sulcus calcarinus. Fibers come to this sulcus calcarinus not directly. They bend in the temporal lobe and then come to this sulcus calcarinus. So it goes. On medial surface, and the same you can draw on this side without. Area 17 by Brodmann. Primary visual center. It is called sulcus calcarinus. Primary visual center, area 17. Primary visual center. Visual sensation, here the analysis of impulses. Our eye could not see nothing. Not in the eye we can see. We see here and after analysis with the brain. And all this is conducting apparatus. Subcortical center, and here Meyer described Meyer's loop. It is called Meyer's loop. And this radiatio optica, optic radiation. It is also by outer graciolate. Graciolates. Radiatio optica. On temporal lobe, this occipital continuous temporal, da? During epilepsy, at patient clinic, perform some part of the temporal lobectomy. Very careful must be with this part. If they cut this Meyer lobe, not only epilepsy can be treated, but also part of the visual fields can be lost. Because Meyer lobe, so through the temporal lobe goes. It is called genicular striate, like stria, lamina, white matter fibers, pass through the frontal temporal lobe to the occipital lobe, sulcus calcarinus. Some part of fibers come from here, from subcortical centers, from tract. To what's subcortical centers? Nuclei tectalis. The same and opposite side, nuclei tectalis. In the tectum, we have gray accumulation of gray matter. It is nuclei tectalis, tectal nuclei. Here lie aqueduct. Under flow of aqueduct on gray matter, we have oculomotor nucleus on both sides. Oculomotor nucleus, and it has not only motor but also accessory nucleus. Yes? Motor and accessory. It is mixed nerve. What pair? This is third pair. Under the flow of aqueduct, we have these fibers, this will come from this nucleus, come here and here to the same and opposite side nucleus. Motor and parasympathetic. Those portions that come to parasympathetic nucleus from here, this side, will go to opposite side, not to the same. To the opposite side will go near the optic nerve. What ganglion lies? Ganglion ciliare. Interrupted here. What name has this portion of oculomotor nerve? It is called radix oculomotoria, oculomotory root. Oculomotor nerve has radix oculomotoria, which is ended on ganglion ciliare. And postganglionic fibers pass inside the eyeball, innervate musculus sphincter pupillae and ciliaris muscle. Slightly below we have what nucleus on the inferior cortical level? 4th pair. Below we have 6th pair nuclei. The 4th, 6th in the brain stem. They are all connected by means of reticular formation fibers, ascending, descending fibers. The same will be on this side. Fasciculus longitudinalis medialis connects 3rd, 4th, 6th pairs of the cranial nerve with nucleus of the accessorius nerve. We can turn our neck and head to the side of light rays, to the object. And this fasciculus longitudinalis medialis. Due this fasciculus is achieved coordinated movements of the eyeballs. All muscles of the eyeball, they are innervated 3rd, 4th, 6th. All together coordinated their movements by means connecting fibers. Fasciculus longitudinalis medialis. It is reticular formation, non-specific tract which connects all these fibers. You can see at the same time that we can see, at the same time, every moment we move our eyeball, do this coordination of fasciculus longitudinalis medialis. At the same time, we can narrow and widen our pupil and accommodation performed through this innervation. And dilatator pupillae receives on the neck region, we have ganglion cervicale superior. Sympathetic trunk ganglion, ganglion cervicale medius, inferior. They come to the superior, and postganglionic fibers will come to the musculus dilatator pupillae. Sympathetic trunk centers are located in the spinal cord. In this intermedio lateralis nucleus, which fibers pass with the spinal nerves and then separate from centrum ciliospinale. Vegetative nervous system, when we describe, we say that this is ganglion cervicale superior. Thus, we can not only see but also perform pupillary reflex accommodation, and pupillary reflex is achieved through sympathetic and parasympathetic innervation. You understand? It is visual analyzer scan. Now hearing analyzer. Hearing analyzer. Scheme of the hearing analyzer. I will draw section of the pons. What peduncles? Cerebellaris media. Pedunculus cerebellaris media. Так, let's draw this. Modulus. and around we have cochlear duct. And here we have lamina tectoria or organ of Corti. And here we have ganglion spirale, spiral ganglia. The dendrite where ended come to this organ of Corti. The axons pass from here and form nervus cochlearis. Another color. The dendrite come to this organ of Corti. Here ended. And from the cells impulse pass here. Here we have organ of Corti inside. Organ of Corti lies here. Dendrite come here, axons leave. Under what name? 8th pair of the cranial nerve. Versibulocochlear, this cochlear portion. Leave the cochlea. And impulses from here come to the pons. Through what foramen come? Through porus acousticus internus, pons where located? On clivus. What nuclei here we have? Usually green color. Do you have a green color? No vegetative. The blue color, it is true sensory. Trigeminal sensory or 7-pair sensory test. But vegetative also, yes, green color. But on this picture, green color will be cochlear nuclei. Nucleus cochlearis. What ganglion is this? Ganglion spirale spiral. It lies in spiral along the modulus. This is modulus. Ganglion spirale cochlea. Which one did you? This. They are bipolar cell. You can draw separately. Separately draw these bipolar cells. They are two poles. Their dendrite goes to this organ of Corti. Their axons here leave the porus acousticus internus. Porus acousticus internus leave, and these fibers come to what nucleus?
Nucleus cochlearis.
Dorsalis and nucleus cochlearis ventralis. We have two nuclei: cochlearis dorsalis, cochlearis ventralis. Nucleus cochlearis dorsalis. Nucleus cochlearis ventralis. The first neuron where are located? In this ganglion spirale. Ganglion spirale, for example, this. The first neuron here, ganglion spirale, bipolar cells. The second neuron are here. Nucleus cochlearis ventralis of this analyzer. Once more, receptors, well located receptors, organ of Corti. Here appears impulse. Dendrites sent to the cell's body, and from axons pass to the synapse and other cells, second neurons. Now their axons of the dorsal neuron, they pass along the rhomboid fossa and then inside and go to the opposite side. And the same will go to this side and go to the opposite side. Axons of the ventral cochlear, nucleus cochlearis ventralis, directly pass to the opposite side. These dorsal nuclear axons, they pass on the dorsal surface of the rhomboid fossa. What structure form on the dorsal surface rhomboid fossa? Which divide the rhomboid fossa. Rhomboid fossa, what structure lie in the center?